So our next technique is FPLC. We will study this technique later in our subsequent lectures. So the next technique is electrophoresis. Electrophoresis is basically used for separating DNA on the basis of its size. So in electrophoresis what we do is कि हम एक जेल बनाते हैं that is called the agarose gel और हम उसके ऊपर लेफ्ट साइड पे एक लैडर लोड कर देते हैं that is used for the allocate different sizes of the DNA of the gene fragment और हम उसके बाद डिफरेंट वेल्स में 1 2 3 4 अपना जीन सैंपल लोड कर देते हैं. फॉर एग्जांपल इफ आवर जीन सैंपल 1 इज एट दिस साइज, नंबर 2 इज at this size, number 3 is at this size, and number 4 is at let's say this size. तो हम ये apparently हम ये कह सकते हैं कि the gene in the sample 4 is smaller as compared to 3 is smaller as compared to 1 is smaller as compared to 2. So हम ये कह सकते हैं कि 2 is the biggest gene among all. हम ये कह सकते हैं बिकॉज दिस रिप्रेजेंट्स द लेटर रिप्रेजेंट साइजेस. फॉर एग्जांपल कि ये अगर लेट्स सपोज 500 KB का है तो ये 400 KB का है, दिस वन इज ऑफ 300 KB, 200 KB एंड सो ऑन. ऊपर 600 KB, 700 KB, 800 अप टू लेट्स से 1000 KB. तो आप अंदाजा लगा सकते हैं कि दिस इज अराउंड 500 KB, दिस वन इज ऑफ लेट्स से 700 KB, this one is of let's say about 350 KB, and this one is around let's say 150 KB. Although the gel is not so big, you can just say it is around. I mean, we can get the idea of how much of the size of the DNA actually, and also the detection of the DNA. We will study this in our subsequent lectures in detail actually. So our next technique is the U visible spectrophotometry. So in spectrophotometry, we will use how different rays of different wavelengths of light are used to detect different biomolecules. For example, if it is a sugar, for example, if it is a protein, if it is a DNA, and if it is an RNA, it will absorb different wavelengths of light. और उस बेस पे कि एक DNA, एक RNA और एक प्रोटीन कौन सी वेवलेंथ ऑफ लाइट को एब्जॉर्ब कर रही है, हम ये बता सकते हैं कि इस स्पेसिफिक लेट्स से इस स्पेसिफिक सैंपल में कितना डीएनए प्रेजेंट है, कितनी प्रोटीन प्रेजेंट है और कितना आरएनए प्रेजेंट है. एंड इवन ये भी बता सकते हैं कि इसमें आरएनए, डीएनए और प्रोटीन प्रेजेंट है कि नहीं. वी कैन ऑल्सो टेल इफ फिनोल्स आर प्रेजेंट, वी कैन ऑल्सो टेल इफ सम एल्कोलॉइड्स आर प्रेजेंट. हम ये सारी बातें जस्ट बाय यूजिंग यूवी विजिबल स्पेक्ट्रोफोटोमेट्री बता सकते हैं कि एक सब्सटेंस में, एक सोल्यूशन में कितने बायोमोलिक्यूल्स प्रेजेंट है और कौन कौन से बायोमोलिक्यूल्स प्रेजेंट है और कितनी अमाउंट्स में प्रेजेंट है एक्चुअली. सो दैट्स हाउ यू विजिबल स्पेक्ट्रोफोटोमेट्री वर्क. उसके बाद है यूज ऑफ रेडियो आइसोटोप्स. सो वी विल ऑल्सो डिस्कस इट इन द दिस इज अ लॉन्ग डिस्कशन, सो वी विल डिस्कस इट इन द सब्सिक्वेंट स्लाइड. सो फर्स्ट ऑफ ऑल हमें ये जानना जरूरी है कि there are many laboratory ethics that we have to be very careful about them while working in a lab. तो इसमें कौन कौन से आते हैं. नंबर 1 इज द मैनर्स ऑफ द लैब. सो एक लैब में काम करते हुए द फर्स्ट एंड फॉरमोस्ट थिंग इज यू हैव टू बी कंप्लीटली प्रोटेक्टेड. आपको अपनी प्रोटेक्शन से ज्यादा अजीज कोई चीज नहीं होनी चाहिए while working in a lab. सो प्रोटेक्शन क्या है. यू वेयर अ लैब कोट. when required you wear gloves. अब मैंने when required क्यों कहा, because sometimes the gloves are not necessary. For example, if you are working with flame, so gloves are very dangerous sometimes, because for example हमारे साथ ऐसा हुआ था when we were in our MPhil कि one of my fellows was working with flame while wearing gloves. the ये तो प्लास्टिक के ग्लव्स होते हैं सिलिकॉन टाइप ना. तो हुआ क्या कि द ग्लव कॉट फायर और वो ग्लव जो है वो उसके हाथ पे लग गए थे. इट वाज वेरी हॉरिबल टू सी दैट. सो use gloves only when required. So when the gloves when actually are the gloves required? So while working with bacteria, while working with viruses, while working with sensitive uh, let's say sensitive biological species, sensitive biological molecules, you use the gloves where where we have to be very stringent about the contamination that can occur in the experiment. So that's when you use the gloves. Okay. But glasses when required. So you get most of the ideas while working in the lab, or lab instructors आपको ये बता देते हैं कि when you will be using glasses, when you will be using gloves, and when you will be using, oh, so lab coat is a must. You will always have to use lab coat in the lab. Okay. So except that the reality is different than movies. अब इसकी एग्जांपल इस तरह लेते हैं कि when we watch a movie, तो हम देखते हैं कि the driver is turning the steering so fast कि ऐसा लगता है just for the physics of action, तो ऐसा लगता है कि it's very, it's very eye pleasing कि हाँ यार वो बड़ी तेज चला रहा है, and असल में ऐसा नहीं होता. So you have to be very careful while driving. Okay, मतलब असल में ऐसा नहीं होता कि you have to move steering so fast just to take one turn. असल में ऐसा कुछ नहीं होता. So it is the same as in the labs. So ऐसा नहीं होता कि जैसा आपको मूवीज में नजर आता है कि just put these chemicals fastly और ऐसे फटाफट काम करें, और ये ऐसा we don't create panic in the lab. So we have to work in the lab very very calmly, very very with very attention, with very care, while being attentive, with very very much concentration. So asal mein, working in a lab is different as compared to what they show in the movies and dramas and TV as well. And the next one is troubleshooting the problem. We will be discussing that later in our in this lecture actually. The next one is cleanliness. So I have a little bit told you about the cleanliness and the safety rules and manners. Cleanliness is like cleaning your bench after you work. It's just as animals do, and even animals clean themselves after they done their stuff, right? So biologists should also be very careful about the cleanliness of the lab. So there are many things that have to be very careful, that we have to be very careful about. For example, if you are working with bacteria, so you have to clean your bench before working with the bacteria, and you have to clean your bench after working with the bacteria. So why are we very careful about these things? Because bacteria are very sensitive things. They are very sensitive. So even one bacterial cell can replicate. एक बैक्टीरिया अगर आपकी कल्चर प्लेट पे गिर गया, तो आपका सारा एक्सपेरिमेंट, month long experiment could go to waste. So we have to be very very careful about cleanliness in the lab and our experiment while working on different projects. So laboratory instructors are very skilled people that help you in almost everything that you are dealing with in the lab. So आप क्या करते हैं कि आपको कोई भी प्रॉब्लम आए, you ask the lab instructors. Don't feel yourself कि आपको इस चीज को let's say चांस लेना पड़ेगा to check it, or to let's say you try to fix it yourself. You just don't try in the lab. You ask the instructor, and for example, if he wants you to learn, then he'll allow you to try. For example, you have आपने DNA निकालना है from the blood. Okay, so you have the blood of a patient that is been suffering from hepatitis C virus. ठीक है? So for example, the blood has been drawn out of the syringe, and you have to be very very careful about the blood. आप उसको उस पे चांस नहीं ले सकते. You have to be very careful about that. ठीक है? You have to be carefully put that into the tube and then spin it to separate the blood, the serum and everything. But you have to be very careful while playing with human blood. So that's how important it is to have lab instructors around while dealing with sensitive experiments. Using equipment such as razor blades and scissors. So another example, the previous example also suits in this point as well. Using of syringe in the lab. So sometimes you have to draw blood. You don't have to draw blood. The certified uh, technicians draw blood from the patient, and they hand over you. And sometimes because of the hectic routine or get many samples, वो आपको सिर्फ syringe लगाते हैं to just pour the blood inside the tube. So you do it with very care, because that is a patient. Okay, so you just don't play with the needle that has been touched with the patient's blood. उस needle के साथ आप नहीं खेल सकते जिसके ऊपर patient का blood लगा हुआ है. So आपको बड़ी carefully उस blood को draw करना पड़ता है. Drawn it's already drawn, yes, but आपको बहुत carefully उसको tube में डालना पड़ता है. That is further used for the separation and whatever the experiment is. Okay, so you have to be very careful about the plate. Using inflammable chemicals. So I have already talked about this. Inflammable chemicals should be dealt with much care, more than the usual one. Okay, because sometimes हमारा ध्यान नहीं होता और आग के करीब हम let's say कि we are performing a bacterial culture, we just have to, हमें सिर्फ ये करना है कि हमें एलबी मीडिया के अंदर कल्चर डालना है एंड वी हैव टू शेक इट और कल्चर ऑटोमेटिकली ग्रो करेगा गिवन द सर्टेन कंडीशंस. पर प्रॉब्लम क्या होता है कि बिफोर क्लीनिंग द बेंच वी हैव टू लेट्स से आवर वी हैव टू यूज फ्लेम टू स्टेरिलाइज आवर इंस्ट्रूमेंट्स. गलती क्या करते हैं बच्चे कि वी हैव टू क्लीन आवर सेल्स विद एथेनॉल एज वेल. बच्चे क्या करते हैं कि एथेनॉल से क्लीन करते हैं साथ ही दे गो फॉर द फ्लेम without taking any care, and the gloves and the hands they catch fire because of the alcohol, because alcohol is highly flammable. So that's what I'm talking about. Okay, we have to be very careful about the flammable chemicals. Okay, the next one is biosafety. So most of the points have been discussed before. The viruses and bacteria, we have taken 2 or 3 examples of that. I think that is no more examples are needed for to discuss such biomolecules. So the next one is the use of UV in biochemistry. So UV ultraviolet rays बहुत important होती हैं biochemistry lab में काम करने के लिए, because even chlorophyll can be studied using UV. So chlorophyll, being green in color, gives a specific color under UV. So we can check if we can check the, let's say we can check the concentration and the quality of the chlorophyll using UV. और इसके अलावा हम जब DNA को स्टडी कर रहे हैं, जैसे मैंने पहले बताया कि different wavelengths are absorbed by different biomolecules. So प्रोटीन को चेक करने के लिए, RNA को चेक करने के लिए, DNA को चेक करने के लिए we can use UV rays. So UV is very harmful for the body as well. इसकी एक एग्जांपल ये है कि when UV जब हमारे स्किन पे फॉल करती है, it can make, फॉर एग्जांपल अगर मैं एक DNA का स्ट्रैंड बनाऊं, तो दिस इज AGCCTTAGC और इसके कॉम्प्लीमेंट्री वुड बी TTGGAATCG. तो नॉर्मली दिस इज हाउ अ DNA स्ट्रैंड इज. होता क्या है कि व्हेन यूवी फॉल्स, तो ये जो बॉन्ड है, ये जो आपको नजर आ रहा है, तो ये बॉन्ड टूटता है और ये बॉन्ड इस तरह से 2 T's के दरमियान बन जाता है. दिस इज कॉल्ड TT डाइमर और थायमिन डाइमर. सो दिस इज अ very big cause of a specific type of skin cancer that is called xeroderma pigmentosa. So just because of some lack of care while using the UV, we can end up in a very difficult situation and very harmful and life-threatening situation. For example, the skin cancer case. So UV को बहुत ध्यान से यूज करना पड़ता है, and UV has many roles in different diseases that we will study in our cell signaling course, inshallah, later in the classes. Next is carcinogenic chemicals. Somehow some chemicals such as ethylium bromide that is mentioned here are very carcinogenic. तो उनको बहुत केयर से इस्तेमाल करना पड़ता है, because those chemicals can cause cancer in your body and even outside the body, just like skin cancer. So उनके बारे में बताया गया. Next is radioisotopes. We will study that in further classes. And then is ethylium bromide. So ethylium bromide is not edible. Sometimes it is very tempting to, let's say, taste the ethylium bromide. However, ethylium bromide is a chemical that is used to visualize the DNA molecule. So, but ethylium bromide क्या करता है? We will study that in our electrophoresis lecture. But ethylium bromide is used to visualize the DNA. So it is very harmful for the body, and we have to be very careful while dealing with ethylium bromide. You will see ethylium bromide lying everywhere in the biochemistry labs when you go to work into the lab. So हमें इसे बहुत केयर से इस्तेमाल करना पड़ता है, and we have to be very careful while dealing with ethylium bromide. Even इसके fumes can be very harmful to our body when we open the bottle, और इसके fumes अगर हमारी nose में जाए. So in that case, what we do is we use masks and we take the bottle very far away from our nose so that the fumes don't go inside, right? So the next one is solving a scientific problem. So I think we will do this in next class of this lecture. So next part of this lecture. So I think we will stop here, and you can ask me questions in my mail or even in the comment box, and we will be happy to answer. Okay, thank you.